Today, we're going to talk about immunity. But before we can understand immunity, we need to understand what is an antigen. Now, an antigen is any foreign molecule that can initiate an immune response. It does not always initiate an immune response, but it is able to. Now, most antigens are proteins or polysaccharides. Polysaccharide refers to a complex sugar. Now, these molecules are often found on the surface of pathogens and cells, like viruses, bacteria, fungi. They can also be found on toxins that are released by pathogens, also on any foreign material. Anyone that's ever had hay fever would know that grass and pollen can initiate an immune response, and then also on our cells themselves. Especially on cancer cells, we get foreign antigens as well. Now, our body is able to produce antibodies. Now, antibodies, which is also called immunoglobulins, are Y-shaped proteins that are produced by our lymphocytes in response to the presence of antigens. Now, the job of these antibodies are to bind to antigens and help the immune system neutralize or destroy the invader. Antibodies are made up of 4 different protein chains. 2 heavy chains, those are the 2 long ones in the middle, and 2 light chains, which are the shorter chains on the sides. They then have these variable regions on the tips of the Y. Now, this part differs between different antibodies, and they can bind specifically to one antigen. These are also called the antigen binding sites. Both of these antigen binding sites will be identical to each other, and they're very highly specific, kind of like a lock and key, where only a certain key can fit into a certain lock. Now, when an antigen is present in the body, it will be able to bind to this antigen binding site because the shape of the antigen is complementary to the shape of the binding site. Now, how are these antibodies made? Now, our body has lymphocytes, which is a type of white blood cells. We have many different lymphocytes, each having different receptor cells on its cell membrane. These receptor cells will be specific in shape to a pathogen. There might be random shapes, there might be shapes that they've encountered before, but it will be specific to a shape of an antigen. When a pathogen enters our body, the pathogen will have an antigen on the cell membrane of the pathogen. When the pathogen encounters a lymphocyte, the lymphocyte will recognize the antigen and bind to it. It will then produce many antibodies that will be able to attach to many more pathogens by binding to their antigens. Now, let's say that there's a pathogen in our body. This pathogen has specific antigens on its cell membrane. Our antibodies will be able to connect and bind to these antigens, making it impossible for the pathogen to move around. This is called agglutination. The antibodies can then also release chemicals that would initiate a process called phagocytosis. This is when a cell called a phagocyte engulfs the pathogen by surrounding itself around the pathogen and then digesting it using digestive enzymes that are found in its cytoplasm. So we can now put this whole immune response into perspective. So first we have a lymphocyte. When a pathogen enters the body, the lymphocyte would recognize the pathogen and then start a process called clonal expansion, meaning that it will replicate itself into many more lymphocytes. These lymphocytes will have receptor cells on their cell membranes that would recognize the pathogen that entered the body. Some of these lymphocytes will produce antibodies for agglutination and the initiation of phagocytosis, and some of these lymphocytes will be made as memory cells to be kept in the body for when the pathogen enters the body again. So that is when a person gets reinfected. This brings us to another part of immunity. We get a primary immune response as well as a secondary immune response. Our primary immune response is when we encounter a pathogen for the first time. We can see in this graph initial exposure. Here the concentration of antibodies are 0. So we have no antibodies that are complementary to the pathogen. So no antibodies will be able to recognize the pathogen. So here the pathogen first needs to be recognized by a lymphocyte to produce new antibodies. We can see the primary immune response is slow, which we can see by the steady slope of the graph. Then, when the immune response that first happened decreases, it never goes back to 0. The reason for this is because memory cells were produced that have the antibodies for the pathogen. We then get our secondary exposure. This occurs when we get infected with the same pathogen. Here our body already has memory cells that can recognize the pathogen. So the secondary immune response is much quicker and much rapid. We can see that more antibodies are produced at a faster rate because the gradient of our graph here is steeper and it goes up to a higher point. This means that the pathogen can probably be eradicated before we even show symptoms. Now we get 2 types of immunity. We get active immunity and passive immunity. Active immunity occurs when we encounter a pathogen. Our immune system, so our lymphocytes, recognize the pathogen. They create antibodies as well as memory cells. This is a long-term immunity, but it's also a slow-acting immunity because our immune system first needs to recognize the pathogen. Now, there's 2 ways that we can get active immunity. The first is by being infected by a pathogen, and the second way is through vaccination. Then, on the other hand, we also get passive immunity. Now, in passive immunity, there's no immune response. So no pathogen is being recognized, no antibodies are being produced, and also no immune response is being produced. So what happens here is that our body gets antibodies from somewhere else. One way is through antibody transfusions. So in this procedure, antibodies are either collected from a person or they were made in a lab and you then get injected with these antibodies. These antibodies can then also bind to the pathogen in your body and it can agglutinate the pathogen and signal for phagocytosis. But no memory cells are made. So although this is a quick response to the pathogen and to the disease, it does not give any long-term immunity. So the immune response after a while will disappear. The second way that you can get passive immunity is as a baby through breastfeeding. The mother's milk contains some of her own antibodies, and she can give these antibodies to her newborn baby through breastfeeding. The same response will happen as in the antibody transfusion. The antibodies can bind to the pathogen and destroy the pathogen, but unfortunately there's no long-term or permanent immunity.